Hi, everyone. Welcome back to today's podcast. In this episode, we're covering Chapter 8 of our textbook, Neuroscience Fundamentals for Rehabilitation by Lori Lundy-Ekman. So, in this episode, we're walking through development of the nervous system. This is from the very beginning of prenatal development all the way through infancy and into the major developmental conditions that come out of this process.
As you listen, keep one big idea in mind. Early development establishes structure. Later development refines function. We'll come back to that idea repeatedly, especially when we talk about why some neurological problems don't show up right away.
So, to understand nervous system development, we need the broader context of prenatal development, which happens in three stages. First is the pre embryonic stage from conception through day 14. And during this time, the fertilized egg divides as it travels to the uterus. Then, during implantation, when the developing organism embeds in the uterine wall, the inner cell mass becomes the embryonic disc. At this point, the disc has two layers, the ectoderm and the endoderm. Next comes the embryonic stage from day 15 through the end of week 8. This is when organ formation occurs, and it's the most critical period for nervous system development. Disruptions here are more likely to cause major malformations. Then, beginning in week 8, we enter the fetal stage, which continues through birth. The focus now is growth and refinement. Structures are already present, but they're becoming more organized and more efficient. This is also when myelination begins, and that process will continue well beyond birth. So, right away, timing matters. Early stages create a blueprint, and then later stages determine how well that blueprint works.
So that blueprint starts with the cell layers. They're layers that differentiate into specific tissues. There are three of them. The ectoderm is one that we care most about here. It forms the nervous system, sensory organs, and epidermis. Every neuron you'll ever study comes from the ectoderm. The mesoderm forms muscle, skeleton, dermis, and circulatory and excretory systems. And the endoderm forms internal organs like those of the digestive and respiratory systems. As these layers differentiate, they also start interacting, and that interaction is what allows the nervous system to form.
Once those layers are established, nervous system development begins in earnest with neural tube formation, which occurs between days 18 and 26. So, a region of the ectoderm thickens to form the neural plate, and then that plate folds inward, creating the neural groove. And when the edges of the groove fuse together, the neural tube is formed. That tube eventually becomes the central nervous system, the brain, and the spinal cord. Closure starts in the future cervical region and it proceeds upward and downward. The open ends, which are called neuropores, normally close by about day 30. And when this process doesn't go as planned, the results can be significant, and we'll come back to that later.
So, as the neural tube is forming, another important population of cells appear, the neural crest cells. These cells separate from the neural tube and migrate throughout the body. They form much of the peripheral nervous system, including sensory neurons, autonomic neurons, and Schwann cells, which later produce myelin in the peripheral nerves. Inside the neural tube, organization is already beginning. By about day 26, the tube differentiates between two concentric layers. The inner layer contains neuron cell bodies and eventually become gray matter, and the outer layer contains neuronal processes that become white matter. The basic gray matter, white matter organization shows up again and again in adult neuroanatomy, as you guys already know.
And at the same time, the adjacent mesoderm divides into somites. These are segmented blocks of tissue that line the embryo from head to tail. Each somite splits into three parts. The sclerotome forms the vertebrae in the skull. The myotome forms skeletal muscle, and the dermatome forms the dermis of the skin. These terms matter clinically. A myotome refers to muscles primarily innervated by one spinal nerve root, and a dermatome refers to skin innervated by one spinal nerve root. Inside the neural tube, farther specialization is happening. We have the ventral motor plate that gives rise to lower motor neurons, which directly innervate skeletal muscle. And we have the dorsal association plate that forms interneurons and projection neurons, which process and relay information within the central nervous system. One developmental detail explains a lot of adult anatomy. After the third fetal month, the vertebral column grows faster than the spinal cord. This is why the adult spinal cord ends around L1 to L2, even though spinal nerves continue below that level.
So while the spinal cord is organizing caudally, brain formation begins rostrally around day 28. The hindbrain develops into the medulla, pons, and cerebellum. The midbrain retains its name throughout development, and the central canal narrows to become the cerebral aqueduct, connecting the third and fourth ventricles. The forebrain divides into the diencephalon and then the telencephalon. The telencephalon then becomes the cerebral hemispheres. As those hemispheres expand, especially as the temporal lobes form, they curve into a characteristic C shape. That's why the structures like the lateral ventricles and the caudate nucleus are also C-shaped in the adult brain.
So during the fetal stage, development shifts toward refinement. Some lateral areas of the hemispheres grow less than the surrounding areas, creating the insula, which lies very deep within the lateral sulcus. And at the same time, the surfaces of the cerebral and cerebellar hemispheres begin to fold, forming the gyri, the ridges, and the sulci, the grooves. Folding increases surface area, which allows for more complex processing without increasing the skull size.
So once large-scale structures are in place, development continues at the cellular level. Neurons undergo differentiation, meaning they develop specialized characteristics like neurotransmitter type and connectivity based on where they end up in the brain. Each neuron extends an axon tipped with a growth cone. The growth cone is dynamic. It senses the environment and guides the axons toward its target. When the growth cone reaches its destination, synaptic vesicles form and microtubules extend to the presynaptic membrane, allowing synaptic communication to begin. Innervation also shapes muscle development. Muscle fibers can actually change type depending on whether they're innervated by fast or slow lower motor neurons. And importantly, early development involves overproduction. The nervous system initially makes far more neurons than it needs. As many as half are eliminated through normal developmental processes, usually because they fail to establish effective connections.
Another major refinement process is myelination. It begins around the fourth fetal month and it continues into early childhood, with most myelin sheaths completed by the end of the third year of life. Because different systems myelinate at different times, abilities emerge in a predictable sequence. Basic sensory and motor skills come first, and then higher level coordination and executive functions come later. And this leads to a key clinical concept called growing into a deficit. Damage that occurs early may not be obvious right away. It becomes apparent instead when the affected system is expected to function. This connects directly to critical periods, which are windows of time when the nervous system is especially sensitive to specific input. And disruption during a critical period can lead to long lasting functional changes, which is why early intervention and timing matter so much.
So now let's talk about infant reflexes and why it matters. Early in development, neck and vestibular reflexes are prominent because cortical systems are still immature. These reflexes are driven by brainstem and subcortical pathways, which develop earlier.
Now, as the cortical systems mature, they begin to modulate and inhibit these early reflexes. So, in typically developing children and adults, the same sensory input is still used, but it's smoothly integrated into voluntary postural control. When early reflexes remain beyond the expected developmental window, it suggests to us that there's some impairment in cortical inhibition. And that's why these reflexes can reappear after extensive cerebral lesions as well. When cortical control is lost, these earlier brainstem reflexes can be released. And for therapists, reflexes provide insight into central nervous system integrity, a level of neurological control, as well as functional motor organization.
So, from a developmental perspective, the CNS is most vulnerable to major malformations between day 14 and week 20. Before this window closes, disruptions are more likely to affect the structures. Afterwards, any insults or injuries tend to affect function, even when anatomy looks relatively typical. This can help explain why individuals with early brain injury can present so differently depending on when the injury occurred.
So there are several major conditions that result from disruptions in neural tube closure, going back to the neural tube. First is anencephaly, and this occurs when the cranial end of the tube fails to close, resulting in absence of the cerebral and cerebellar hemispheres. We also have Arnold-Chiari malformations, which involve deformities of the hindbrain. Type 1 Arnold-Chiari malformation is often asymptomatic, and type 2 presents in infancy, and it may involve hydrocephalus and cranial nerve dysfunction. Then there's spina bifida, which results from failure of the inferior neuropore to close. Spina bifida occulta is hidden, and spina bifida aperta includes meningocele, myelomeningocele, and myeloschisis. I encourage you to look in your textbook to see pictures of what this looks like. Then there's tethered spinal cord, which occurs when the cord adheres to the lower vertebrae, restricting movement and causing dermatomal and myotomal deficits, something called saddle region pain, which is pain in your saddle region, and bowel or bladder dysfunction. Symptoms may not appear until adolescence if the traction on the spinal cord is mild.
And there are some other conditions that reflect disruptions at different points within the development. You have spinal muscular atrophy, or SMA. This involves degeneration of lower motor neurons due to deletion of the survivor motor neuron 1, or SMN1 gene. Fetal alcohol syndrome results from parental alcohol exposure and affects cognition, behavior, and motor function. Cocaine exposure. Cocaine exposure most commonly disrupts neuronal proliferation. Errors in neuronal migration can cause heterotopia, often associated with seizures. And intellectual disability is frequently associated with abnormalities in dendritic spines, which are key sites of synaptic input.
So, cerebral palsy is a group of permanent, non progressive movement and posture disorders that are caused by damage to the developing brain. Because the injury occurs during development, not after skills are acquired, the movement patterns reflect disrupted development rather than a loss of function. So, the types of cerebral palsy include ataxic cerebral palsy, hypotonic cerebral palsy, spastic cerebral palsy, dyskinetic cerebral palsy, or mixed cerebral palsy. How many times can you say cerebral palsy? Importantly, most cases result from prenatal events, not birth trauma.
And then we also have abusive head trauma in infancy, which can cause severe disability or death and may resemble cerebral palsy. There's developmental coordination disorder, which involves impaired motor coordination despite normal intellect. ADHD is associated with inattention, impulsivity, and hyperactivity, along with differences in frontal, striatal, and cerebellar regions. And then autism spectrum disorders involve impairments in social communication and restricted behaviors. And extensive research shows there is no link between autism and the MMR vaccine or Tylenol in pregnancy, for that matter.
All right. So early development establishes structure, while later development refines control, integration, and efficiency.
Oops, I took a drink of water, sorry.
I'm just going to leave all this in. See what happens.
When disruption occurs, outcomes depend on when, where, and how development was altered.
So, understanding nervous system development provides essential context for assessment, for intervention, and for long term clinical reasoning, especially when working with children and individuals with early brain injury. Okay, see you next time. Thanks for listening. Bye
Bye. Bye.
bye.